compare and contrast between some of the early events in the animal development that we've been talking about throughout the course of the semester, and how life is for plants. So of course plants do have developmental biology, they have embryo stages, and they have a lot of the same sort of life history challenges that they're going to have to meet, just like animals do. But they're also different in their morphology, so they may need to take a somewhat different approach in some cases. So the first major difference in plants versus animals is there is no gap relation in plants. So, in animals, once we reach this multicellular stage after fertilization, there is this massive rearrangement of all these tissues, and that established the three germ layers: ectoderm, mesoderm, and endoderm, which we now appreciate is really a fundamental way to organize this body, and it gives rise to all these different downstream organs and tissue types and cell phases. So plants, because these cells are surrounded by cell walls, these tissues are just simply not able to move in the same way that animal cells do. So they're going to need to take their existing structure and sort of in the status quo, divide it up into different territories and assign the cell fates based on that. So there's another somewhat quirky difference from plants, and you may remember back from intro bio, I'm sure this got discussed, but plants have this sort of bipartite life cycle known as alternation of generations. So they have some of the point where they're a diploid organism, so 2N. They're obviously multicellular during this stage, and this is known as the sporophyte. Then they're going to take some of the cells from that sporophyte and send those cells through meiosis. So the products that you get out of that are known as spores. They're haploid. And then as haploid cells, they will divide by mitosis. So once again, you establish a multicellular organism composed of entirely haploid cells. Okay. So now these spores are going to be known as the gametophyte, and after going through mitosis and becoming multicellular, some of those haploid cells will become the gametes, sperm and egg, and then those participate in fertilization for the next generation. So we'll talk about this in more detail today. But just kind of think of this in two categories: a yellow category, a blue category, where you're diploid for part of the life cycle and haploid for part of the life cycle. So also just to point out here that when these diploid cells are going through meiosis, this process in animal makes our gametes, but in the plants, the products of meiosis are the spores, not the gametes. Gametes don't come until you have become multicellular, gone through mitosis, and then some of these haploid individual cells will be specified as the gametes. So that's a separation of that cell fate from meiosis. Okay, at least for some of the animals, like C. elegans and Drosophila, we said that they have set aside certain cells very early in development to become the germ cells, sperm and egg. And plants are different because they do not do this. They wait until they have an adult structure, and then some of those cells at that point will be specified to become sperm and egg. Another thing is we've been talking the entire semester about embryo life and embryo development, and really the only thing that was not embryo development was just the last lecture where we talked about the secondary sexual characteristics happening at puberty. But in plants this is different, and what you'll see is the morphogenesis is going to occur over really the entire life of the plant. So morphogenesis in plants would be including making things like additional shoots, additional leaves, roots, flowers. All of those structures are not created in an embryo and then get bigger, but rather you get this plant that is built, and then from meristematic tissues within the plant, you have these new structures that grow out. So that's something that just happens, over the life of this plant and repeated repeated cycles. So what this means then is that meristems are going to be these regions of adult stem cells within the plant, and they're very important for the continued ability of that plant to grow and form additional structures. So they'll be persisting in that plant long after we can call the plant a mature flower, trees, or whatever. Oops, I hit the wrong button. Okay, so another theme from animal development is that these cell fates have become increasingly restricted as development proceeds. So in our zygote, we had, the ability to give rise to any and all daughter cell fates that you see in the organism, and then this becomes more and more restricted. And cells have to eventually make decisions, go through commitment, turn on specific gene products, and then finally, as a result of all of these decisions, they will begin to show their structural and functional characteristics. And this paradigm for cell fates also needs to happen in plants because they have a number of different differentiated types of cells. Some of these are going to be leaf cells, others would be flower cells. So they also need to go through these same challenges of deciding how do you assign a cell fate to a particular cell and how do you regulate differential gene expression within cells. And one contrast to animals in this regard is that plants tend to maintain a very high status of pluripotency for most cells, even in the adult state. So animal cells, in the embryo may be still pluripotent, but usually by the time you've gotten out of the embryo stage and you're moving on to, say, larval development or childhood, those cell fates are pretty much established. And now we appreciate we could take these cells and put them in culture and maybe express some genes like the IPS genes and cause them to reverse it. But in their natural endogenous state, that's not going to happen. They've already made some decisions early on, and that's not going to be reversed too much. But in plants, they kind of hang on to this ability to maintain their high pluripotency or even totipotency, even for just your ordinary cell in a plant stem. And so this means that it's pretty easy to manipulate these plants in culture, much more so than animals. And you can do things like take little sections of plant tissue, put it on an agar plate with some hormones, and just grow kind of a mass of generic cells known as callus, which is shown here. And then you can take those cells, whatever they used to be, now they're much more generic in nature. You can add back the appropriate hormones and stimulate those cells to regrow as more shoot-type tissue or more root-type tissue. And you can have a relatively easier time to cause these single plant cells to regenerate into whole plantlets that have both leaf and apical portions and root portions as well. So there's a lot of interest in trying to do this for various different types of plants, including some of our crop plants or just other plants of economic interest, and seeing what we can do to manipulate the genetics of these things using tissue culture tools. So this plasticity or this flexibility, if you will, on the part of plants comes in kind of handy for those types of studies. Okay, another thing is that animal cells, you'll typically see they may be haploid, they may be diploid, but they don't have a very high tolerance for anything other than that. So it's pretty rare to get conditions where you have triploidy or even aneuploidy, and that's compatible with survival. So aneuploidy, just to remind you if you're not familiar with this term, would be that you have an extra chromosome or you're missing one chromosome. So like let's say within your normal diploid set, everything is okay, but chromosome 21 is now found in 3 copies. That would be an aneuploidy situation. Okay? or you're basically diploid for every chromosome, but you're happen to be missing one of chromosome 11. That also would be aneuploidy. Polyploidy, to contrast this, is going to be looking at full sets of chromosomes. So haploid, diploid, triploid, tetraploid, and so forth, where you're adding not just one chromosome, but the entire haploid set would be part of that nucleus. So in general, you can find examples of polyploidy in nature with animals, especially with, say, some fish are tetraploid, and they manage to handle that, but it's not that common. And plants on the whole are a lot more tolerant of this status, and so people have, over the years, bred this and gotten some interesting polyploid situations. So red wheat, we have 6-fold ploidy. Rye is actually 2, and you can breed these together and get an 8-fold ploidy intranucleally, and this seems to do okay for these cells. They can go through meiosis and mitoses normally. And then what about the developmental mechanisms? So we've heard all about these Hox genes and master regulatory genes and reciprocal induction. Thinking of different paradigms we've talked about over the course of the semester, to what extent do these apply also to plants? And it varies. So sometimes you'll see similar sorts of things, and other times plants seem to have evolved a different way to meet the challenge. So in animals, we talked a lot about homeobox genes, just as a family that contains the conserved sequence of the homeobox. And you will find some homeobox genes in the plants. So they definitely do exist there, and they have roles in shaping morphogenesis and patterning, so in that sense, there's a similarity there. However, in animals, it's really these homeobox genes, especially the Hox genes, that set up the body plan along the anterior posterior and specify these different regions of identity along that body axis. And in plants, that's really not the case. We'll talk tomorrow about a different group of genes known as the Mansbach genes that have a conserved region in our family. And it's that set of genes that seems to be mainly responsible for patterning the meristem and kind of dividing up that territory and assigning different cellmates to it, so that some part will become a leaf, some part will become a petal, or as the case may be. So the mechanisms that plants use for patterning, so in one sense they're still relying on transcription factors and conserved families of them, and actions in early development to divide up a territory, so that's kind of similar, but then the actual gene family is different. Okay, and just in common, I mentioned some of this already, but of course plants do have development. They have embryonic and adult stages. They are going to make gametes. They go through fertilization. So they're going to face some of the same constraints, maybe even more so, since they can't get up and walk around. They need to make sure that they're able to sense, is this gamete coming in of the correct species? How can I attract a gamete so that appropriate numbers of sperm are available to fertilize my eggs? How can I disperse my gametes into the local environment? All these sorts of natural constraints are issues for plants, and they have to find mechanisms to deal with those so that they can continue the species. And then once we do get a successful fertilization, we're still going to have to go through all of these major challenges of getting the appropriate numbers of cells, putting them in the right place, getting them to cooperate with each other so that you can get some pattern and morphogenesis, and then also differentiating the appropriate numbers and types of cells. Okay, so to look in more detail about what happens for the life cycle of plants in early development, I want to really focus in this class on angiosperms. So angiosperms are a class of plants that are the flowering plants, and there are other life cycles of other groups of plants, and they do it somewhat differently, but I think the main points come through just by looking at angiosperms, so we're going to focus on them, but just to point out at the get-go, so the full disclosure that this is only one subset of the plants that are out there. Okay, so angiosperms are going to have, as we said, the two different generations, as will all plants. And in the case of angiosperms, it'll be the sporophyte that is the dominant leafy generation that we know and love. So when you go out there and look around and see some flowering plants, you're looking at sporophytes, or plants composed of diploid cells. In the case of some things like mosses, it may be actually the gametophytes that is what we're looking at. What we think of as moss is actually the gametophyte generation, and all that structure is composed of haploid cells. So that's one factor that can vary. So here in the sporophyte generation, we are going to see fertilization occur, and once we get an embryo, that will reestablish our diploid sporophyte generation. This embryo will grow over time into the large leafy tree, plant, flower, or whatever. Then within the flower, we're going to need to sequester two separate territories that are going to make male gametes and female gametes. So this is another thing that is kind of characteristic for angiosperms, is that they do this. They have a separate portion of tissue that makes male gametes and one that will make female gametes, you know, down the line once we get to this part. But for right now, in the flower structure, we can point to microsporangium up there as a region that will give rise to these things called microspores that eventually make the male gametophyte. So now in plants, this prefix micro is going to refer to all of the structures leading to male development, sperm down the line. Whereas mega will refer to all of our female structures. So there's going to be kind of a lot of vocabulary in today's lecture, but just keeping that micro and mega separate will be a first step. Yeah.
Is there a big difference in size like there is for animals in the male and the female spores?
Somewhat, yeah. Yeah, so you can get these large starter eggs with a large cytoplasmic volume, that's for sure. Okay, so up here is anther. So in the purple there we see our flower parts where we're going to have these stamens composed of long filaments with anthers on top of them, and within the anther, that's where you're going to see your diploid cells that head through meiosis and make the microspores. In the middle, we have this kind of tan structure that is called the carpel, and this is where you're going to have the cells that give rise to the female lineage. So there's a region called the megasporangium, and this will be where meiosis occurs to generate the female gametophyte. So then once we get over here to this stage after meiosis, we're going to have haploid cells, microspores or megaspores, that then go through meiosis and make a male gametophyte or a female gametophyte. So these are both multicellular structures, and they're separate individuals. So one individual is a multicellular male gametophyte, another is a multicellular female gametophyte, and then these guys will go through then mitosis, and make the sperm and egg that eventually fertilize each other into restoring the sporophyte generation. Okay, so let's look at these two basic processes within the megasporangium and the microsporangium in a little bit more detail. So in the flower, angiosperm, we have the anther, and up here you see this cross section through the anther. And some of these cells are somewhat similar to mesenchymal cells, or your basic loose arrangement of cells within the plant. And these are known as tapetum. So the outside cells that compose the walls of these structures are diploid cells, and they're providing kind of an environment where we can have these pollen sacs. Pollen sacs are also called the microsporangium. And by the way, some of these are Latin terms, so you're going to get your nice Latin endings. IUM. This is usually a singular form of the world, so microsporangium. And IA would be the plural form, the microsporangia. So within the microsporangium, this will be cells are going to start going through meiosis here. And eventually we'll get mature pollen, which is actually the male communiphyte, these microspores that develop. So here's the process. Up here we have a cross-section through the anther, and we're showing 4 pollen sacs or 4 microsporangia. And within these, we can find individual diploid cells that are called the microspore mother cell. So these are the cells that will undergo meiosis. So meiosis 1 and 2. They're going to make the 4 meiotic products. All of them survive. So here are 4 different haploid cells with their chromatin stained pink, so you can kind of see where the nucleus are in these cells. And they're actually still kind of held together because they've been dividing from one microspore mother cell. So this structure is known as the pollen tetrad, and it contains the 4 meiotic products. And these cells individually are called the microspores, and they're haploid. Then these haploid cells, each one of them, will now undergo mitoses. So they go through one round of division and make these 2 cells. One of the cells is called the generative cell, and one is called the tube cell. So when we make a pollen grain, it'll be the tube cell that is the cell, it's actually a single cell that grows an entire tube that grows down the length of the plant in the female part and carries the sperm to the egg. So once we get to this now multicellular, it's not very elaborate multicellular, it's only 2, but still that's more than 1, so we count it. This multicellular individual is going to do something kind of strange, and that is that this generative cell basically gets swallowed up into the tube cell. So now you are going to have the tube cell entirely existing within the cytoplasm, the generative cell entirely existing within the cytoplasm of the tube cell. So here it is, this is the nucleus and the cytoplasm of the generative cell. Inside of the tube cell, here's the tube cell nucleus and the tube cell cytoplasm. Then the generative cell will divide one final time, and that gives rise to 2 sperm cells. So here's your generative cell, its nucleus, its cytoplasm, haploid cell, goes through mitoses, again. Now we have 2 sperm cells already existing within the cytoplasm of the tube cell. So this is what the pollen looks like. This is the male gametophyte, and also up here, the pollen, if we look at these grains under the microscope, you can often see these really beautiful and elaborate protrusions and little hairs sometimes. And this gets to the pollen function in life, which is to be dispersed across the landscape by wind or insects or sticking to genes on humans, human clothing, getting carried and moved around the environment and brought to the new plant. So they're specialized for that purpose. And these outside coatings are then secreted by the tube cell. And they need to be sure that the pollen grain can survive some harsh environmental conditions. So there's often kind of waxy substances. And then also make sure that the pollen grain gets appropriately dispersed so that it can ultimately land on another plant individual and germinate and then deliver those sperm cells to the egg of that new individual. Okay, so question?
Yes, I'm just trying to, so it's pollen can be both the microspore and the microgametophyte, just depends on if it's gone through mitosis once. Or it's a microspore when it's by itself, and as soon as it gets one round of mitosis, it's also now considered a microgametophyte.
Yeah, so these terms are kind of crazy here. So there's a pollen sac, and then we're going to make the pollen tetrad, which has 4 microspores in it. And then, so far these are just microspores. They need to go through a couple of rounds of mitoses, first to generate the 2 cells, and then another one to generate the 2 sperm cells. And it's not really until we get to the final stage here of the 2 sperm cells within the generative cell, that thing is the pollen grain.
And that's considered the microgametophyte?
Yes, that's considered the male gametophyte, not the microgametophyte. Male gametophyte. So all of these structures, anytime since from the minute you get to be haploid, you're a male gametophyte. And then you're gonna, doesn't really matter how many cells you have. First you'll be a single cell, and then you'll be multiple cells, and then eventually you'll be a pollen grain. All that whole time you're still the male gametophyte. So basically everything on the blue side of the equation there is male gametophyte. Okay. Good question, good thing to think about, wrestle with. Okay, so here is just a slide showing you again a cross-section here in anther. And this anther has some mature pollen within it and is now undergoing a process of degeneration of these tapetal cells, and that will release the pollen to the environment. So what happens to it? This pollen grain is going to go over and hopefully land on a stigma of either the same plant. In some cases, some plants are able to make pollen that can fertilize their own stigma. In other cases, they have to fertilize the stigma of the same species but different individuals. And it just depends on the plant whether or not one or both of those situations can happen. So this is the center of the plant here where we see these structures known as carpels. And it includes a sticky region at the top called the stigma, and this is often specialized to try to let the pollen grains that land on it adhere, and then it provides some moisture to the pollen grains so that they can germinate and begin to grow down the long length of that female structure, and this is called the style. Then this gray structure here is a pollen tube. That's all part of that single tube cell that's just extending, 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 and getting very, very long and narrow. And within the carpel here, we have ovules, which are structures that would contain the female gametophyte, including the egg. So these pollen tubes have to find the little opening in an ovule so that they can bring the sperm cells down and introduce it into the female gametophyte and allow fertilization of the egg. So this shows you an expansion of the tip of the pollen tube with the tube nucleus, so that cell still has its nucleus present, and then the 2 sperm cells that contain both cytoplasm and nucleus and plasma membrane and all that good stuff that ordinary cells usually contain. So they're right there in the tip, and they're getting brought down to find the egg and participate in fertilization. So just to kind of think about all of the different kinds of cells that are involved in this process of bringing the sperm to the egg and allowing fertilization to occur, we have some cells that are sporophyte. So these are just the structure of the flower that was in the yellow part of the diagram, the structure of the flower that was going to contain the female gametophyte, and the style cells are diploid. And then these pollen tubes, these are part of the male gametophyte. They came from the pollen. They're haploid, and they're growing down, down, down. They contain the haploid sperm. And then eventually we'll get to the female gametophyte, which is called the embryo sac, and that's a haploid multicellular structure. So we have some cells haploid, some cells diploid, and everything having to interact with each other. So then any genes that are expressed in any of these different cells might influence the successful function of those cells in terms of making sure fertilization happens smoothly. So we could ask, what is the mechanism of making sure that that pollen tube could actually find the opening and allow the sperm cells to be delivered to the egg. And this has been an interesting question because it has to do with how well plants are going to be able to be successful in their reproductive stages. So it does have some economic concern. But also just from the point of view of basic developmental biology, how do plants get the job done, we'd like to understand what mechanisms are involved. And one thing that probably plays some role is these calcium channels that are found right at the tip of the growing, extending tube cell within its plasma membrane. And there seems to be a wave of calcium that is extending along the length of this pollen tube and may play some role in the successful continued growth or directionality of that pollen tube. So that's one possible thing. In addition, there's two cells next to this blue egg cell here within the female gametophyte that are thought to secrete a chemical attractant. So a chemotaxis type of mechanism attracting the pollen tubes to grow, specifically to this little opening here in the ovule, is probably another widely used mechanism to make sure that pollen tube gets down to that opening. Okay, so here are the different portions. I probably should have put this slide earlier, but you should be familiar with all of these different terms. For stamens are composed of the anthers and the filaments. The carpels are composed of stigma, style, and ovary. And then individual chambers containing female gametophytes are known as ovules. So an ovary may have one ovule or maybe several, depending on the plant. And then the basic structures of plants, that they have these a few small leaf-like structures at the base of the flower. Those are known as sepals. Then they'll have a layer of petals, a layer of anthers, and then a layer of one or more carpels in the very center of the plant. So that's your basic angiosperm